Good morning, everyone.
Good morning.
Hi, hi, hi. All right. Okay, I think we have 2 or 3 papers today. If you guys could share it in the chat. Here we go, more people coming. Sorry, I switched off the sound. Here we go. Right, Victor's paper is already in the chat. I think everyone presenting today knows how to do it. There was one person who sent me an email that she wanted to present today, but I can't see her in the room yet. If she joins, we take her in. Other than that, you know how it works. Just briefly present your paper, tell us why you're excited about it, and we have a chat about it. Given that the other two who may or may not have a paper haven't sent the link yet, Victor, why don't you start us off?
Sure. All right. Okay, here. All right, can you see the screen correctly? Perfect. So the article that I found this week was recently published in Neuroimage, or at least, I don't know if it is online on their website at least. It's called Deep Learning Based Unlearning of a Dataset Bias for MRI Harmonization and Confound Removal. This is quite interesting to me because, well, it's working with deep learning algorithms that are very, well, a la mode in the field of neuroimaging in a lot of different, for a lot of different uses. And it tackles a problem that is quite that is quite an issue with a lot of small data sets or of cross studies use of deep learning. What I mean is, what it does is trying to improve the prediction. Okay, I think I'll start from the start rather than from the middle. So globally, there is a lot of deep learning algorithm that tries to use the data from MRI, whether it be like raw data or pre-processed data, images, or even just metrics of connectivity metrics extracted from the data. And so they use this data and they train an algorithm, a deep learning algorithm, to predict some other variable. Typically, for example, we could use the images, the T1 images from MRI scan of a cohort, and to try to predict the age of each subject using only the T1 scan. It could also be other modalities, but just bear with me. So there is, with deep learning, we need first, well, at least with some class of deep learning, I will just stay simple. We need we need to make the algorithm learn from learning data sets the relationship that there is between the input image and the output age. So you need to know both at the same time. But then when it has learned the relationship between them, you can use new data that the algorithm has never seen and ask it, what will the age be of each subject in the new data set? And it works really well, not in every kind of variable. Age works quite well depending on what you input in. I think sex gender really works very well, but it can also be used to try to predict the pathological states of some people, and it works also pretty well in this case. But one problem is that usually when you train an algorithm on one data set, it will learn also all the biases of this data set. Meaning, for example, you train an algorithm on the, let's say, the UK Biobank or the the UK Biobank. We train an algorithm to predict the age on the UK Biobank, and it will work great if, for example, you train it on half of the UK Biobank, and then test it on the other half that it has never seen, and you'll see that on the other half, it will give you a nice prediction of the variable that you were searching for. And then you want to apply this algorithm, this trained algorithm, to the let's say the HCP dataset. Thing is, those are two very different datasets with different modalities, and they used different machines to acquire them. They use different processing pipelines to process the data. So there will be ever so slight differences that you might not see with your bare eyes, but that the algorithm will have picked up and will not be able, will use in as information in the prediction that it made. So this is a problem with this kind of algorithm. And now that we have all of this, to come back to the article, what it tries to do is to remove the information learned by the algorithm that is linked to the what they call the domain, meaning the all that is linked to the specific data set that you were using at first. The different data set. For example, it would be the domain can comprise the scanner, the pipeline, and etc., and everything that is linked to it. And so to do this, they use a classical scheme of deep learning. Like here, it's so also for right now they just try to remove the effect of the domain on the output. And only do it for one specific task. It's not something that you do generally. When I mean one specific task, is it will only work on the algorithm that learn to predict one specific variable. So you'll need to do every time you need to predict another variable and everything. So for example, if you what they call you unlearn the domain in the algorithm, and you do it for the age prediction, you cannot just apply what you did on the age prediction to the sex prediction. You have to redo the old procedure in the algorithm that learned to predict the sex. But that's still very interesting to do it as they do. So what the algorithm does is it takes the input data and try to predict both the variable that you want, so what they call the main task, and at the same time the domain classifier, the domain in which the data was taken in. So you need at least two domain in your training data set, meaning you have to have, for example, in a study with multiple sites in which the data was acquired, you need to give the site as an input variable to the classifier, and it will try to predict which domain, so which site the data was acquired in, and knowing that, it will then try to relearn everything while confuse, they say that they try to confuse the domain classification. Basically, they try to make so that the domain classification is the worst possible, meaning that the classifier, the algorithm cannot, well, only takes the information that is relative to the task and not at all the one that is relative to the domain. And so one good example, I think, that they give here, let's see, can you see if the image when I do this? Okay, so is to use, like here, the biobank, the UK Biobank dataset and the OASIS dataset, and they train the algorithm on several different little fields, well, little domain, well, space where there is, sorry, I'll go back to this. So they train the algorithm on the age domain that is shared between the two datasets, so that they can have a comparison between each subject, saying, okay, this for the algorithm, you'll have a subject that is when during the training that is 20, well, here, well, maybe more like 60 in the OASIS dataset, and one that is also 60 in the biobank dataset. So the algorithm will be able to compare and to learn the differences that there is between the two datasets. And so you do the training on this small band here of age, and they did it for several size of overlaps of ages to check how it works, if the more the overlap is and if it's better and everything. But this is the idea, so that the algorithm can learn what not to look at when it's trying to learn the age or another variable. So let's get to the last point that I wanted to show. Okay, and so this was the principle, and here you have an interesting result. They give the mean absolute error of the results when comparing ABIDE, another dataset, the ABIDE dataset that uses several different sites to acquire the data, and they used so the error of prediction. They compare the error of prediction if they do the simple learning by the algorithm, or the one with the unlearning of the site of the different site of the ABIDE dataset, and they compare it. So in light blue, it's the normal classical prediction, the error of prediction. So the higher the better, the worse. And in dark blue, it's with the unlearning algorithm. So you can see that sometimes it's comparable and not that great to use the unlearning on some of the sites. So here it's a different site that the task that I was acquired in this axis. But on some of the actually in a lot of the sites, it really improved the prediction of the data to use the unlearning algorithm. So it's still a first step. It's in this kind of field. I'm quite new to this. I don't. There's probably a lot of things that already have been done in here in this field, but to me, it's something that is really interesting and that needs to be more addressed. That I haven't, at least myself, hadn't heard of before, and it's nice to know that there is ways to remove this kind of information when you try to apply a deep learning algorithm to do some cross dataset prediction, and also to make, for example, your algorithm, if you make a deep learning algorithm, to say that, okay, it was trained on this data set first, but then we improved it so that it can be used by people using the HCP dataset 2 and the UK Biobank dataset 2 and a lot of different datasets with the trained model that we use here. So usually in the world of deep learning, you give the algorithm and say, okay, now you can train it on your dataset, but that means that it's limited. You have to have lots of data and you need to retrain everything. Here with this kind of thing, I think in the future we can ship directly trained algorithm that you just need to be applied on new dataset, hopefully, and get results immediately. And that's, I think, a good thing for the future.
Thank you, Victor. It's very interesting paper. Definitely above my head a little bit, to be honest, this morning. Oh yeah, question, Michel. Oh, he found a new button. Look at that.
Yeah, yes.
So it's really interesting. I was wondering if if you push this thinking to the furthest, if you apply machine learning to kind of normalize your data set and make them comparable, do you think there is a potential danger? It's kind of like applying a correction lens in the way you're looking at data. And do you think this correction lens can bias our observation in any way? Do you see what I mean? That's a potential caveat.
What?
That's a potential danger.
Oh yeah, caveat. Okay.
My accent from Liverpool.
To apply this kind of correction without thinking and to just think there is no bias. Well, when we
I'm not talking about the center bias. I'm talking about applying a filter, a transformation to your data using machine learning that you don't control completely, but might eventually make you lose some information. Just because it works like a smart filter somehow.
Yeah, I see what you mean. That's not easy to say. I think it's not such a big problem in this particular case, because what we are trying to do is to remove a lot of the existing bias. I mean, you can always say since you don't know exactly what you are removing, and that's true. With deep learning, it's quite a big critic that we can hear a lot of time. You don't exactly know what you're doing with the data. It's a black box that you created. You know how the black box is made, but you don't know what it learns. That is usually true, although there is a lot of stuff. There is a lot of work being done here, trying to better understand what's going on inside of it.
But look, for example, the KKY and NYU, like those two data sets have been acquired on two sides of the planet, and they're definitely going to have differences that will be considered biased, but are like true difference across populations. For example, these kind of things, how do you think the algorithm can handle that?
Well, that's the whole point of the algorithm, right? It's to just find what the difference are between the two data sets and to ignore them. It's not trying to understand them. What I can say, one caveat that
I see it's great, but in the precise case here, KKY is, I think it's from China, and NYU is from United States. Some of the difference are like true phenotypes that change the functioning of the brain. If you normalize that,
all right,
you put this lens. So like you're gonna all be the same with my lens, but the truth is it's not entirely true.
Yeah,
you see what I mean?
Yeah, I completely see. Okay, now
I'm
I understand.
not saying it's not a good method, but I'm trying to see both sides.
No, that's actually an excellent point. I didn't think of that because I was already thinking, it should be about the same person that goes in different missions. But no, you're right. When it's at two different sides of the world, you have different populations, and so it will remove this aspect of the data. And that is this is very interesting to take into account. And Well, that also means that you have to be careful when you apply this kind of thing. But still, I think if it's done intelligently, should be okay. But yeah, you're right. We really must be careful with that.
Open question.
No, it's true. It's yeah, good, very good point.
I will always fight the machine.
Why are you saying this?
You know,
all right.
Thanks for that discussion. Are there any other questions? Yes, Leah.
Thank you, Victor. It was really interesting. And like connecting to your discussion, I was thinking, so if these deep learning algorithms works really well in the prediction, as you show, of age, for example, like if each side want to participate to this project, so like how many subjects did they say in the article, how many subjects did they need to do a training and their validation? They give like a general idea, because if it's dangerous to apply, for example, the algorithm developed from the ACP dataset to your dataset, there is also the possibility to develop your own deep learning algorithm if is it so working well. I don't know in pathology, but like for healthy people, if is it working? Generally, no.
So yeah, your question is that, if I understand correctly, that if you have a small data set and you cannot train on it really, can you apply something that was trained on several data sets and just put it on yourself?
No, if you can, you have your own data set,
yeah.
and you want to apply the training, the deep learning training that they validated on the ACP, for example, but you want to do everything again in your data set. So, it is not the point. Yeah, maybe
okay,
not the point.
Okay, all right. So the thing is, well, it's a great question. Yeah, usually you need a lot of data to do this kind of retraining, but because it's trying to use data from both the big data set and smaller data set at the same time, and just remove the domain information, they say that you can use very small data set. For example, it's what they did here with the different sites in the Abide dataset. It's the Abide, the Abide dataset, and they said that on some sites you only had 27 subjects, which is extremely few for deep learning. Usually, you need hundreds of subjects to have a good prediction on your data. So, I think
indeed this seems feasible.
It's feasible. I'm quite surprised that it works well with so few subjects, but it seems that it's one of the strengths of this technique. I need to check it more thoroughly because I haven't had the time to read all of the small details. But it's it seems to be working this way.
Okay, yeah, I think this is really a good point. Thank you.
Thank you for your question.
You don't have to read all the small print for Neuroccino. That's
the whole point. That's why I did not.
But well done, well done. I got one question that you may or may not have actually covered, but I, for example, had the age of my brain estimated before and after I did my PhD. I know it's terrible. I aged 6 years in 3. But still younger than my actual age, so not too disappointed with that. But there was a certain error rate associated with that, which I think at the time was ±4 years. So I was wondering what the error rate here would be, and how that, if that would be the same across data, for example. Would that matter?
Specifically for the age, I have no idea, and the error rate really depends a lot on the data you put in and the variable that you're trying to predict. For example, I tried to apply deep learning to predict data, to predict the age on connectivity data, and it was not really good. And I saw articles that were trying to predict the age on T1 datasets, and they were really doing some pretty good. They had pretty good results, but still with an error rate, an error margin that was quite high. Age is not the easiest thing to predict, at least not right now with the method that we have, and also with the variability in how the brain age and what the difference between true age and the brain age and lots of stuff like that. So the error in the prediction, at least in the age prediction, I cannot really answer your question here. It's something that we have to deal with, and in this particular example, don't remember. I think they give some values, but I don't remember where it is.
Don't worry about it. We can read the small print later together. But thank you. Whilst our next speaker is preparing coffee for us here, given that it's Neuroccino. Perfect. Thank you. Two things I quickly wanted to mention. Number one is that with the new update of Zoom, when you raise your hand, it actually gives me a little summary at the bottom, so I won't miss your questions, which is fantastic. So if you could use that feature to raise your hand, it's really easy. The other thing is we had a couple of issues last week and this week as well with the link. So just FYI, we have changed the link because for those of you were there just before Christmas, we got trolled and we did not want that to continue. So we changed the link, but the link for the session is on the website and it hopefully should work. We can see your screen, Victor, just FYI.
Oops, sorry, I stopped sharing.
Yes, so if you struggle to connect to the meeting or it says the link is not valid, check the website. If it happens again, we will just update the link on the website. So you should always be able to connect. If you use our calendar, you should have an up-to-date link. If it doesn't work, please do let us know and Leah will change the links for you in the calendar. Okay. Now, without further ado, Michelle, whenever you're ready, take it away.
Unmute. There you go. All right, so what happened and is exciting in science last week? We have this paper's been published in Brain Structure and Function somehow last week. It'll be in the next issue of Brain Structure and Function, which is it's a review about the red nucleus structure and function from anatomy to clinical neurosciences. So who knows about the red nucleus? Okay. Well, you know a lot more in a minute. So what's fascinating about this review is like they did it all. They added inside the same manuscript knowledge about different species, put it in perspective with the phylogeny, the evolution of the brain, linking it up with pathologies and future development and questions that we should solve. And that I found that was like a beautiful way to bring things together and cover comprehensively a question about a brain structure. So if we speak about the evolution of the red nucleus, right, as you may know, is inside the brain, of course. It's located right under the thalamus. And if you compare it, if you look at the very primitive red nucleus, there was only a connection between the red nucleus and the spinal cord and dentate and cerebellum. So you can have this cerebellar-spinal relationship that happened. And then as evolution carried on from a very primitive to a lizard, which would be representative of what dinosaurs had, you had these other pathways that started to appear that connect the inferior olive of the brain, which is another nucleus. And then things got like a lot more complicated with mammals, where you had an involvement of the thalamus and cerebral cortex that is also interacting with the red nucleus, that started to segregate itself into two subregions, one with big neurons, which is the magnocellular red nucleus, and one with small neurons inside it, which is the parvocellular red nucleus. And those two sections that divided in the brain of the red nucleus connect differently the circuitry that is connected with the inferior olives from the circuitry that is directly connected to the spinal cord. And so like you see how things are being progressively evolving and how structure change progressively with evolution. So that's great, but you see you get the red and the green pathways. The question is, what are doing those pathways in the brain? So after going through the entire literature and summarizing what we know, which is derived from different species, we know that the magnocellular, which is the red one here, connecting the spinal cord in the brain, the magnocellular primary role is in locomotion and postural stability. Think about when you're getting onto these electric stairs, mechanic stairs, I don't know how you call that. When you're going in the subway, well, you need to adjust your stability. This is where it's happening. And this has been described in quadrupedal animals, mostly mammals, but it disappeared, this relationship, with bipedal primates, monkeys. So they don't use this region for that. And they use this region mostly for hand movement, reaching and grasping. So you see how a structure that was there originally in animals has been recycled for you to optimize your interaction and your manipulation of objects. And that's where, functional evolution has been faster than anatomical evolution and require recycling of systems that were devoted to things that were different for assuming a function that is new and required by that species. And that's all happening in the magnocellular system that you can see here in red. Now, the parvocellular system, because you cannot study it in every animal, like we know much less about it, but what we know about it is that, it's really debated, but it has an importance for controlling the execution of reflex, I don't think it takes plural. So they say including error sensing, timing, and learning of acquired motor behavior, or reflex conditioning. The exact functional role are highly debated and a clear consensus is still lacking, but it seems that it plays a role in the control of this reflex and the execution. But it also goes further than that. And if you study it in primates, you can see that the red nucleus is not only involved in this motor dimension, as we know that the cerebellum is not only motor, but it has also some importance for the pain induction and modulation. So it has a control of this information of pain. And that's interesting, how a structure that seems to be mostly motor, that is starting to take like some cognitive dimension, if we can say that pain is as complex as cognitive. The problem is, like, we don't know much about the human red nucleus. So you have this wonderful table here where they report meta-analysis, they report all the results that have been reported in terms of the connectivity of the red nucleus, trying to decipher and understand really what is the function of the red nucleus in humans. And there's been more recycling of the red nucleus in humans that will be explained by connectivity, I don't know, with the language area or space or things like that. But the prime is like the very, you have very contrasted result, not only due to the method, but to the fact that the red nucleus is a small structure. It's hard to have a pure, neat connectivity profile of this region with the rest of the brain, plus the limitation that we know of the neuroimaging approach into showing really a good picture of the connectivity pattern. So it's kind of like, you know, we think because the red nucleus have been showing this fantastic evolution across species and took like new functions through this recycling approach, we think there is like fantastic things to discover, but they still remain to be explored in order to be discovered. The end of the review is really good as well, because, you know, the red nucleus anatomy and how it evolved, why does it matter? Well, it does matter because the red nucleus is involved in a great deal of pathologies of the brain, starting from essential tremor, where it is like a major participant, but also in Parkinson's disease, where recent finding, as I say, found that the red nucleus, which is rich in iron, show alteration with Parkinson's disease. And then you also have interesting studies have been demonstrated that after a stroke that impairs the corticospinal tract or pyramidal lesions in monkeys, the red nucleus network is taking over some of the motor function, helping for motor recovery. As kind of a shortcut or bypass of the standard network, it can take over the function and help the brain recover from its injury. So a lot of perspective from this structure that are nicely summarized in the conclusion in 6 bullet points that I'll read to you. The first one is phylogenetically, the red nucleus is likely to be correlated with the appearance of limb or limb-like structure, and it plays an important role in the transition from aquatic to terrestrial locomotion. That was from primitive to quadrupedal, primitive to lizard, sorry. Then the primitive red nucleus functionally corresponds to the magnocellular red nucleus and is involved mostly in locomotor function and in the execution of simple stereotype movement. The parvocellular red nucleus developed later and is probably related to increasing behavioral complexity. That's an easy one. Then the third point is also already present in quadrupedal mammal animals. Magnocellular red nucleus functional specialization for upper limb movements become more evident with the evolution of bipedalism, and that's recycling I was talking about. Then it is unclear if the magnocellular red nucleus undergoes a complete regression in the adult human brain, but it is possible that it can play a role during human ontogenesis or in the recovery of pyramidal lesions. That's what I was saying. We don't know exactly what happened with the red nucleus in human with the complexification of the system. Is it like a structure that is left out and is regressing, or did it start like being recycled for new functions? It's definitely a high impact paper there for the person who discovers that. Function of the parvocellular red nucleus are still largely unknown in animal models, but it can be hypothesized that they may control the motor system on a higher level in cooperation with the olivocerebellar and basal ganglia system because they are highly connected. And finally, a new finding in rat model and human neuroimaging implies the role of red nucleus in mediating anti-nociceptive response to pain stimulation. It's interesting how a motor structure suddenly took over pain-related function in the brain. So yeah, that said, I think it's a very good example how you can write a review about a structure or a system in the brain. It is very comprehensive. I found it extremely helpful. Definitely my best reference for the red nucleus to recommend to anybody who would like to learn about it. There is everything in it, from the anatomy to the function to the evolution and the pathology. And that's what makes an excellent review, I think.
Thank you, Misha. It's also quite astonishing. It's completely unfunded from what we can see here. A very
Yeah.
comprehensive work. Any questions from you? Leah, go ahead.
Thank you. This is really interesting again. I was thinking, did they say in the review if high field can help to do a step further in this detection of complex connectivity, for example, with tractography of the red nuclei?
No, they didn't mention that because it's not coming really from the neuroimaging field.
Okay, because I saw the table.
Yeah, they tried to be comprehensive and had humans by collecting every paper that reported the connectivity. But if you want, the limitations that they put on MRI and connectivity approaches made me like think that, well, they don't know how much we limited in our approach with MRI. They really take it and compare it to axial tracing, like, yeah, should be the same.
Okay.
While we actually know that we very, very limited.
Okay.
Especially for small structure like this. But post-mortem diffusion imaging, particularly in a small animal like the squirrel monkey, will help us to decipher the true connectivity of this structure.
Okay, so like all the main findings were from histology or tracing. So not, okay, I get it.
But I mean, if you want to look at the connectivity of the red nucleus, you need to have like diffusion at 100, 200 micron. I think that's the kind of resolution you're looking for to answer this question, with comparable results.
Thank you.
Yeah, my comment was going to go in the same direction actually, but with that table and the DTI, especially with 1.5 and 3 Tesla and a mix between deterministic and probabilistic, and the red nucleus is tiny.
I know. It's I mean, that's all we have, and I salute the effort of trying to summarize all this information about the red nucleus into this table, showing everything that has been done, and it's a work in progress. But I don't think we have the necessary methods yet to answer this question in human using neuroimaging.
But that's also a good learning point, right? Just because a software gives you a pretty picture, it may or may not actually be anatomically plausible and relevant.
That's absolutely right.
Okay, are there any other? Yeah,
I have a question. It's not directly related to this, but more generally, it's if it's possible to link to have a better link between MRI study and small nuclei like this. If you know, maybe if there are studies using, for example, they do only they do the acquisition and the MRI. I don't know if they exist. Well, if they do a acquisition of the MRI only on specific slices where they have, for example, the structure that they want to look at, typically small nuclei, and maybe another part of the brain that they think is related. It is related to, and they do because they would do some only. They would do the acquisition only on some slices. They could increase the resolution drastically. And I don't know if it exists, but just the idea of this that comes to my mind made me think that maybe it exists and it could be used to investigate the functional connectivity of specific brain, small brain structure. And do you have heard of something like that, or?
No, that's a good idea. So the problem is, like, if you don't have the entire brain, you cannot see the profile of connectivity with the rest of the brain. No, but if you could have two MRIs, one that is doing a high zoom onto your region and another one that is doing a higher, lower resolution the entire brain, and you could synchronize the two, that'd be cool.
I don't know if it's possible.
I never heard about it.
Yeah.
Build an MRI inside an MRI.
No, that's probably not possible. But...
No, well, No, I don't know how you send your radio frequencies and that.
Yeah, that would no.
I mean, no, it's really it's really how the antenna is going to collect the information. So if you have two antennas, like it's probably if you have two antennas and they collect in parallel,
yeah,
that could be possible.
We'll have to ask a physicist, right, Leah?
What does a physicist think about it?
I am like in my experience, I saw that the thing that you proposed for QSM for another small structure, the nigral it's also affected in Parkinson's disease.
Substantia nigra.
Yeah, but the small wind that you have nearby that you can say a change in the QSM, and there are some studies that acquire with high resolution only that nuclei or that layers that you're interested, and then it's a good idea, as Michelle proposed, to merge with a more comprehensive acquisition of all the brain. And so I think it's possible what you proposed, Victor.
Oh, that's nice.
I was also thinking that maybe if the radionuclei are so important in maybe rehabilitation or plasticity, maybe with fMRI you can see an indirect measure of what is changing. So if the radionuclei are so small to detect morphologically, maybe you can measure the changing in connectivity in some indirect way. I was thinking about that, but both the ways are, I think, possible. Or if you select few slices and increase the resolution, that because if you increase the resolution, of course, you increase the timing of the acquisition. But if you have a few slices, so that will not be a great problem.
That was my idea.
Yeah. So it's then you can also increase the bandwidth. You can increase other fields. Of course, it will depend on the limit of the gradient, the magnetic gradient fields that you have. So there will be some limits in the scanner that you have to deal with. But if the problem is the timing, if you acquire few slices, of course, you manage this problem in another way. And then you can acquire another fast acquisition and make some comparison, for example.
Okay, thank you.
Sounds like you have a project for the summer now. Right, are there any other comments? No. Yeah, oh, sorry, go ahead.
This is not a real question, and thank you for appreciating the review. I'm among the authors, and I think that
I hope I did it right.
all the comments are really useful and interesting. I want to answer to Leah that we looked into 7 Tesla, and there is the possibility to improve our actual knowledge with this, I think, but there is still a lot of work to do, as Michel said, on this field. It would be interesting to deepen the aspect of structural connectivity and also of functional connectivity, as also Victor said, increasing the resolution, cropping the MRI, it would of great interest. But this is such a tiny structure. Also, in fMRI, it's not easy to disentangle what's happening in those mesencephalic area. So yes, it's still a lot of work to do, but thank you all for the appreciation.
It's an amazing paper. I will make the cover of Brain Structure and Function, by the way, next month.
Very happy.
Right. So I think that's nice final words. We still got a lot of work to do. So with that, I wish you a wonderful week in science, and I'm looking forward to seeing you next Monday again for the next Neuroccino. Thanks for coming, everyone.
Thank you. Bye.
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 Hey guys, I wanted to say hi.
Wait. And
We still-
stream.
Still streaming.